Molecular evolution is an important process to understand as it provides a link between the underlying DNA sequences that make up our genome and the functions that are ultimately encoded at the protein level that give rise to the phenotypes that compose each organism that we're interested in studying. And so it's through this filter of evolutionary processes that molecular changes that happen at the DNA level can affect ultimately how organisms look, how they behave, and how many offspring they have that constitutes their fitness from an evolutionary perspective. The mutational process to genes is a random process, so mutations happen at random with approximately equal chance anywhere within the gene. Then you have this process of fixation where, depending upon what that mutation does, it has a chance of being lost from the population or a chance of becoming fixed within the entire population. So this is what gives rise to the non-randomness of observation of genes, is this process of fixation once a mutation has happened. You can have other types of mutations that give rise to either insertions of new sequence or the removal of sequence that's existed for long periods of evolutionary time in a gene, and so that is something that happens as well. So one question that emerges is how mutation limited is evolution? How much does a population wait for mutations that are beneficial to emerge within a new environment? And that depends in part on the number of individuals, the census size of that population that's moved into this new environment. In some cases where you have a large census size, the mutations that are necessary to adapt to a given environment already exist within the population and can be selected. So as species diverge from each other, one attribute of adaptation is adapting to a new environment. So environments change over time, and species move into new ecosystems, into new environments. And so to enable an organism to survive within a given environment, sometimes genetic changes that are possible will increase the fitness of that organism and make it better able to survive and reproduce, and so to have a higher fitness in that environment.
There is definitely variation in the genomes of individuals within a species. So, for example, there are differences in the genome among different people, and these differences are called alleles. And a lot of what people are interested in right now are characterizing those genetic differences and what aspects of the differences we see among different individuals can be due to differences in their genome.
So there are mutations that are segregating, for example, in the human population in enzymes that enable our ability to metabolize lactose, or in our ability to metabolize ethanol, so alcohol, that are different within different individual humans and are at different frequencies in different parts of the population. And so these changes can function in two different ways. So they can affect either the amount of a protein that's produced, so it's the expression level of the protein, or the actual function of the protein by affecting the rates of catalysis or other attributes of protein function if you're an enzyme.
In fact, this is a big area now in medicine because they're finding more and more that drug responses are a great example. Some types of drugs are very effective in some people and not so effective in other people. And what they found is that they can look at genetic variability among people almost as markers to try and be able to say, okay, if you have these certain markers, they're kind of like signposts in the genome. If you have these certain markers, then you're likely to respond well to this drug. Whereas these other types of people with these different markers in their genome, these drugs might not be as effective for you. And that's, the whole goal of precision medicine now, to try and characterize that a little bit better. It's one of the ways in which human genome sequencing is seen to be useful.
When one undertakes screens looking for positive selection across genomes between closely related species, where we can compare them all in a phylogenetic tree structure, one uncovers large numbers of genes that show evidence for particularly rapid sequence evolution. And several of these genes will show charismatic stories. And so one of these is plasminogen activator in vampire bats, which is linked to blood clotting in mammals. Other examples would include myostatin in ruminant artiodactyls, so in cattle and sheep and pigs, where this protein has also undergone rapid sequence evolution and may be linked to the number of skeletal muscle fibers that the organism has. Other examples include opsin genes and its relationship, for example, in whales and in seals to the depth that the organisms dive and how they perceive light in different environments. And so there are all sorts of stories that can be told that uncover important aspects of lineage-specific biology that we can uncover through these types of screens.
So when we recreate the past at the molecular level in the laboratory, we can actually, and we test its function, we can actually use that information to try and better understand not only organismal biology, but also aspects of the ecology that changes in natural selection that are acting on these genes might be in response to. So, for example, in the visual system in which I work, we try and understand some of the visual environments that organisms might have evolved in by reconstructing components, molecular components of the visual system, and looking to see how their function is different from those components that exist today. And from that, we can infer aspects of the visual ecology of the organism. So to give you an example of a mutation that we have evidence was selected for in the course of evolutionary history, this is some of the work coming out of my lab by one of my students, Sarah Dungan. She is studying the visual system of whales, of cetaceans. And she's basically studying the protein in your eye that responds to light and basically allows you to see. And this is true of whales, it's true of people, it's actually true of all organisms that can see. They use this protein called rhodopsin. So she was investigating the rhodopsin in cetaceans and using some statistical methods to detect which parts of this rhodopsin gene might have been targeted by selection in order to adapt to the underwater world in which whales live, right? Because we know that whales evolved from terrestrial ancestors that at some point in evolutionary history went into the water and evolved this very unique and fascinating, fully aquatic lifestyle. There were lots of sensory adaptations that happened to enable them to adjust to this amazing, aquatic life history, including things like echolocation, but also aspects of their visual system had to adapt. So basically, if you think about it, underwater aquatic environments are very dim and very blue compared to most terrestrial environments. And so these whales had to adapt their visual system to that. So what she found when she did these statistical analyses of these particular visual genes, these genes underlying vision, is that there were certain parts of the genes that seemed to be more important than others that may have been targeted by natural selection. So using these statistical methods, she was able to generate a very strong hypothesis about which parts of the gene might be allowing the whales to adapt to their unique underwater environments. And so then we thought, all right, we have this very interesting hypothesis, but in my laboratory, we can also take those genes and express them in the laboratory and then test aspects of their function in the laboratory. So that's what we did. And then we can also mutate. We can change the parts of the genes that those statistical methods told us might have been the targets of natural selection, and we can see do they actually change the function of this protein to allow it to be able to function better in dimly lit, blue-shifted light environments? And so we were able to show in the laboratory that, in fact, it's true. When you change those parts of the protein, you get better adapted proteins that are more sensitive to light and function better in those aquatic environments.